Organic compounds essential to human functioning. Organic compounds typically consist of groups of carbon atoms covalently bonded to hydrogen, oxygen, and often other elements as well. They are found throughout the world, in soils and seas, commercial products, and every cell of the human body. The four most important types to human structure and function are carbohydrates, lipids, proteins, and nucleic acids. What makes organic compounds ubiquitous is the chemistry of their carbon core. Recall that carbon atoms have 4 electrons in their valence shell, and that the octet rule dictates that atoms tend to react in such a way as to complete their valence shell with 8 electrons. Carbon atoms do not complete their valence shells by donating or accepting 4 electrons. Instead, they readily share electrons via covalent bonds. Commonly, carbon atoms share with other carbon atoms, often forming a long carbon chain referred to as a carbon skeleton. When they do share, however, they do not share all of their electrons exclusively with one another. Rather, carbon atoms tend to share electrons with a variety of other elements, one of which is always hydrogen. Carbon and hydrogen groupings are called hydrocarbons. Many combinations are possible to fill carbon's 4 vacancies in its valence shell. Carbon may share electrons with oxygen or nitrogen or other atoms in a particular region of a compound. Moreover, the atoms to which carbon atoms bond may also be part of a functional group. A functional group is a group of atoms linked by strong covalent bonds that tend to function in chemical reactions as a single unit. 5 functional groups are important in human physiology. These are hydroxyl groups, carboxyl groups, amino groups, methyl groups, and phosphate groups. Hydroxyl groups are polar. They are components of all 4 types of organic compounds. Carboxyl groups are found within fatty acids, amino acids, and many other types of acids. Amino groups are found within amino acids, which are the building blocks of proteins. Methyl groups are found within amino acids. Phosphate groups are found within phospholipids and nucleotides. Carbon's affinity for covalent bonding means that many distinct and relatively stable organic molecules readily form larger, more complex molecules. Any large molecule can be referred to as a macromolecule. Some macromolecules are made up of several copies of single units called monomers. These monomers are linked together by covalent bonds to form long polymers. Monomers form polymers by engaging in dehydration synthesis. This reaction results in the release of one molecule of water. Each monomer contributes to this process. One gives up a hydrogen atom, and the other gives up a hydroxyl group. Polymers are split into monomers by hydrolysis reactions. The bonds between their monomers are broken via the donation of a molecule of water. This water contributes a hydrogen atom to one monomer and a hydroxyl group to the other. Carbohydrates. The term carbohydrate means hydrated carbon. Recall that the root hydro indicates water. Thus, a carbohydrate is a molecule composed of carbon, hydrogen, and oxygen. In most carbohydrates, hydrogen and oxygen are found in the same 2:1 relative proportions that they have in water. In fact, the chemical formula for a generic molecule of carbohydrate is CH2ON. 3 forms of saccharides are important in the body: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are the monomers of carbohydrates. Disaccharides are made up of two monomers. Polysaccharides are the polymers and can consist of hundreds to thousands of monomers. Monosaccharides. A monosaccharide is a monomer of carbohydrates. 5 monosaccharides are important in the body. 3 of these are known as hexose sugars. They are called this because they each contain 6 atoms of carbon. These are glucose, fructose, and galactose. The remaining monosaccharides are the 2 pentose sugars, each of which contains 5 atoms of carbon. These are ribose and deoxyribose. Disaccharides. A disaccharide is a pair of monosaccharides. Disaccharides are formed via dehydration synthesis, and the bond linking them is referred to as a glycosidic bond. 3 disaccharides are important to humans. These are sucrose, commonly referred to as table sugar, lactose, or milk sugar, and maltose, or malt sugar. As you can tell from their common names, you consume these in your diet. However, the body cannot use them directly. Instead, in the digestive tract, they are split into their component monosaccharides via hydrolysis. The monosaccharides glucose and fructose bond to form sucrose. The monosaccharides galactose and glucose bond to form lactose. 2 glucose monosaccharides bond, forming maltose. Polysaccharides. Polysaccharides can contain a few to 1,000 or more monosaccharides. 3 polysaccharides are important to the human body: starches, glycogen, and cellulose. Starches are polymers of glucose. They occur in long chains called amylose, or branched chains called amylopectin. Both of these are stored in plant-based foods and are relatively easy to digest. Glycogen is also a polymer of glucose, but it is stored in the tissues of animals, especially in the muscles and liver. It is not considered a dietary carbohydrate because very little glycogen remains in animal tissues after slaughter. However, the human body stores excess glucose as glycogen in the muscles and the liver of the body. Cellulose is a polysaccharide that is the primary component of the cell wall of green plants. It is the component of plant food referred to as fiber. In humans, cellulose, or fiber, is not digestible. However, dietary fiber has many health benefits. It helps you feel full, so you eat less. It promotes a healthy digestive tract, and a diet high in fiber is thought to reduce the risk of heart disease and some forms of cancer. Functions of carbohydrates. The body obtains carbohydrates from plant-based foods. Grains, fruits, legumes, and other vegetables provide most of the carbohydrates in the human diet, although lactose is one found in dairy products. Although most body cells can break down other organic compounds for fuel, all body cells can use glucose. Moreover, nerve cells, known as neurons in the brain, spinal cord, and throughout the peripheral nervous system, as well as red blood cells, can only use glucose for fuel. In the breakdown of glucose for energy, molecules of adenosine triphosphate, better known as ATP, are produced. Adenosine triphosphate, or ATP, is composed of a ribose sugar, an adenine base, and 3 phosphate groups. ATP releases free energy when its phosphate bonds are broken, and thus supplies ready energy to the cell. More ATP is produced in the presence of oxygen than in pathways that do not use oxygen. The overall reaction for the conversion of the energy in glucose to energy stored in ATP can be written as glucose plus 6 molecules of oxygen form 6 carbon dioxide molecules, 6 water molecules, and ATP. In addition to being a critical fuel source, carbohydrates are present in very small amounts in the cell structure. For instance, some carbohydrate molecules bind with proteins to produce glycoproteins, and others combine with lipids, producing glycolipids. Both of these are found in the membrane of the cell. Lipids. A lipid is one of a highly diverse group of compounds mostly made up of hydrocarbons. The few oxygen atoms they contain are often at the periphery of the molecule. Their nonpolar hydrocarbons make all lipids hydrophobic. In water, lipids do not form a true solution, but they may form an emulsion, which is the term for a mixture of solutions that do not mix well. Triglycerides. A triglyceride is one of the most common dietary lipid groups, and the type found most abundantly in body tissues. This compound, which is referred to as a fat, is formed from the synthesis of two types of molecules, a glycerol backbone and 3 fatty acids. The glycerol backbone is at the core of the triglyceride and contains 3 carbon atoms. The 3 fatty acids are long chains of hydrocarbons with a carboxyl group and a methyl group at opposite ends, each extending from the carbons of glycerol. Triglycerides form via dehydration synthesis. Glycerol gives up hydrogen atoms from its hydroxyl groups at each bond, and the carboxyl group on each fatty acid chain gives up a hydroxyl group. A total of 3 water molecules are thereby released. Fatty acid chains that have no double carbon bonds anywhere along their length are considered saturated fatty acids. Saturated fatty acids contain the maximum number of hydrogen atoms. These straight, rigid chains pack tightly together and are solid or semi-solid at room temperature. Butter and lard are examples, as is the fat found on a steak or the fat inside of your body. In contrast, fatty acids containing 1 double carbon bond are kinked at that bond. These monounsaturated fatty acids are therefore unable to pack tightly together and are liquid at room temperature. Polyunsaturated fats contain 2 or more double carbon bonds and are also liquid at room temperature. Plant oils, such as olive oil, typically contain both mono and polyunsaturated fatty acids. Whereas a diet high in saturated fatty acids increases the risk of heart disease, a diet high in unsaturated fatty acids is thought to reduce that risk. This is especially true for the omega-3 unsaturated fatty acids found in cold water fish, such as salmon. These fatty acids have their first double bond at the 3rd hydrocarbon from the methyl group, referred to as the omega end of the molecule. Finally, trans fatty acids found in some processed foods, including some stick and tub margarines, are thought to be even more harmful to the heart and blood vessels than saturated fatty acids. Trans fats are created from unsaturated fatty acids, such as corn oil, when chemically treated to produce partially hydrogenated fats. As a group, triglycerides are a major fuel source for the body. When you are resting or sleeping, a majority of the energy used to keep you alive is derived from triglycerides stored in adipose tissue. Triglycerides also fuel long, slow-paced physical activity, such as gardening or hiking, and they also contribute a modest percentage of energy for vigorous physical activity. Dietary fat also assists the absorption and transport of the nonpolar fat-soluble vitamins A, D, E, and K. Additionally, stored body fat protects and cushions the body's bones and internal organs, and acts as an insulation to retain body heat. Fatty acids are also components of glycolipids, which are sugar fat compounds found in cell membranes. Lipoproteins are compounds in which the hydrophobic triglycerides are packaged in protein envelopes for transport in body fluids. Phospholipids. As its name suggests, a phospholipid is a bond between the glycerol component of a lipid and a phosphorus molecule. In fact, Phospholipids are similar in structure to triglycerides. However, instead of having 3 fatty acids, a phospholipid is generated from a diglyceride, which is a glycerol with just 2 fatty acid chains. The third binding site on the glycerol is taken up by the phosphate group, which in turn is attached to a polar head region of the molecule. Recall that triglycerides are nonpolar and hydrophobic. This still holds true for the fatty acid portion of the phospholipid compound. However, the head of the phospholipid contains charges on the phosphate group. These charges make the phospholipid head hydrophilic. Therefore, phospholipids are said to have hydrophobic tails containing the neutral fatty acids and hydrophilic heads containing the charged phosphate groups and the nitrogen atom. Steroids. A steroid compound, also known as a sterol, has as its foundation a set of 4 hydrocarbon rings bonded to a variety of other atoms and molecules. Although both plants and animals synthesize steroids, the type that makes the most important contribution to human structure and function is cholesterol. Cholesterol is synthesized by the liver and is also present in most animal-based foods. Like other lipids, cholesterol's hydrocarbons make it hydrophobic. However, it has a polar hydroxyl head that is hydrophilic. Cholesterol is an important component of bile acids, which function to help emulsify dietary fats. Cholesterol is also a building block of many hormones. Hormones are signaling molecules that the body releases to regulate processes at distant sites. Finally, like phospholipids, cholesterol molecules are found in the cell membrane, where their hydrophobic and hydrophilic regions help to regulate the flow of substances into and out of the cell. Prostaglandins. Like a hormone, a prostaglandin is one of a group of signaling molecules. But prostaglandins are derived from unsaturated fatty acids. One reason that omega-3 fatty acids found in fish are beneficial is that they stimulate the production of certain prostaglandins. These prostaglandins help regulate aspects of blood pressure and inflammation, and thereby reduce the risk for heart disease. Prostaglandins also sensitize nerves to pain. One class of pain-relieving medications, called non-steroidal anti-inflammatory drugs, or NSAIDs, works by reducing the effects of these prostaglandins. Proteins. You might associate proteins with muscle tissue, but in fact, proteins are critical components of all tissues and organs. A protein is an organic compound composed of amino acids linked by peptide bonds. Proteins include the keratin in the epidermis of the skin that protects underlying tissues. Proteins also include the collagen, which is found in the skin, in bones, and in the meninges that cover the brain and spinal cord. Proteins are also components of many of the body's functional chemicals, including digestive enzymes in the digestive tract, antibodies, the neurotransmitters that neurons use to communicate with other cells, and the peptide-based hormones that regulate certain body functions. While carbohydrates and lipids are composed of hydrocarbons and oxygen, all proteins also contain nitrogen, and many contain sulfur, in addition to carbon, hydrogen, and oxygen. Microstructure of proteins. Proteins are polymers made up of nitrogen-containing monomers called amino acids. An amino acid is a molecule composed of an amino group and a carboxyl group, together with a variable side chain. Just 20 different amino acids contribute to nearly all of the thousands of different proteins important in human structure and function. Body proteins contain a unique combination of a few dozen to a few hundred of these 20 amino acid monomers. All 20 of these amino acids share a similar structure. All consist of a central carbon atom to which all of the following are bonded: a hydrogen atom, an alkaline amino group, an acidic carboxyl group, and a variable group. All amino acids contain an acidic carboxyl group and a basic amino group. For this reason, amino acids make excellent buffers, helping the body to regulate acid-base balance. What distinguishes the 20 amino acids from one another is the variable group, which is also known as a side chain or an R group. These groups can vary in size and can be polar or nonpolar, giving each amino acid unique characteristics. Amino acids join via dehydration synthesis to form protein polymers. The unique bond holding amino acids together is called a peptide bond. A peptide bond is a covalent bond between two amino acids that forms via dehydration synthesis. A peptide, in fact, is a very short chain of amino acids. Strands containing fewer than about 100 amino acids are generally referred to as polypeptides rather than proteins. The body is able to synthesize most of the amino acids from components of other molecules. However, 9 of these cannot be synthesized and have to be consumed in the diet. These are known as essential amino acids. Free amino acids available for protein construction are said to reside in the amino acid pool within cells. Structures within cells use these amino acids when assembling proteins. If a particular essential amino acid is not available in sufficient quantities in the amino acid pool, the synthesis of proteins containing it can slow or even cease. Shape of proteins. A protein's shape is determined most fundamentally by the sequence of amino acids of which it is made. The sequence is called the primary structure of the protein. Although some polypeptides exist as linear chains, most are twisted or folded into more complex secondary structures that form when bonding occurs between amino acids with different properties at different regions of the polypeptide. The most common secondary structure is a spiral called an alpha helix. If you were to take a length of string and simply twist it into a spiral, it would not hold its shape. In a similar manner, a strand of amino acids could not maintain a stable spiral shape without the help of hydrogen bonding, which creates bridges between the different regions of the same strand. Less commonly, a polypeptide chain can form a beta-pleated sheet, in which hydrogen bonds form bridges between different regions of a single polypeptide that has folded back upon itself, or between two or more adjacent polypeptide chains. The secondary structure of a protein further folds into a compact three-dimensional shape, referred to as the protein's tertiary structure. In this configuration, amino acids that had been very distant in the primary chain can be brought quite close together via hydrogen bonds, or in proteins containing cysteine via disulfide bonds. A disulfide bond is a covalent bond between sulfur atoms in a polypeptide. Often, 2 or more separate polypeptides bond to form an even larger protein with quaternary structure. The polypeptide subunits forming a quaternary structure may be identical or different. For instance, hemoglobin, which is the protein found in red blood cells, is composed of 4 tertiary polypeptides. 2 of these are called alpha chains, and the other 2 are called beta chains. When proteins are exposed to extreme heat, acidity, alkalinity, and certain other substances, they can denature. Denaturation is a change in the structure of a molecule through physical or chemical means. Denatured proteins lose their functional shape and are no longer able to carry out their function. The contribution of the shape of a protein to its function can hardly be exaggerated. For example, the long, slender shape of a protein strand that makes up muscle tissue is essential to its muscle's ability to contract and relax. Bones contain long threads of a protein called collagen that acts as a scaffolding upon which bone minerals are deposited. These elongated proteins, called fibrous proteins, are strong and durable and typically hydrophobic. In contrast, globular proteins are globes or spheres that tend to be highly reactive and are hydrophilic. The hemoglobin proteins packed into red blood cells are an example. However, globular proteins are abundant throughout the body, playing critical roles in most body functions. Enzymes, introduced earlier as protein catalysts, are examples of this. Proteins function as enzymes. If you were trying to write a paper, and every time you hit a key on your laptop there was a delay of 6 or 7 minutes before you got a response, you would probably get a new laptop. In a similar way, without enzymes to catalyze chemical reactions, the human body would be essentially non-functional. It functions only because the enzymes function. Enzymatic reactions begin when substrates bind to the enzyme. A substrate is a reactant in an enzymatic reaction. This occurs on regions of the enzyme known as active sites. Any given enzyme catalyzes just one type of chemical reaction. This characteristic, called specificity, is due to the fact that a substance with a particular shape and electrical charge can bind only to an active site corresponding to that substrate. Due to this jigsaw puzzle-like match between the enzyme and its substrates, enzymes are known for their specificity. In fact, as an enzyme binds to its substrates, the enzyme structure changes slightly to find the best fit between the transition state and the active state. This active site modification in the presence of the substrate, along with the simultaneous formation of the transition state, is called an induced fit. Overall, there is a specifically matched enzyme for each substrate, and thus each chemical reaction. However, there is some flexibility as well. Some enzymes have the ability to act on several different structurally related substrates. Binding of a substrate produces an enzyme-substrate complex. It is likely that enzymes speed up chemical reactions in part because the enzyme-substrate complex undergoes a set of temporary and reversible changes that cause the substrates to be oriented towards one another in the optimal position to facilitate their interaction. This promotes increased reaction speed. The enzyme then releases the products and resumes its original shape. The enzyme is then free to engage in the process again and will do so as long as substrate remains. Other functions of proteins. Advertisements for protein bars, powders, and shakes all say that protein is important in building, repairing, and maintaining muscle tissue. But the truth is that proteins contribute to all body tissues, from skin to brain cells. Also, certain proteins act as hormones, which are chemical messengers that help regulate body functions. As mentioned earlier, the basic and acidic components of proteins enable proteins to function as buffers, maintaining acid-base balance. Proteins also help to regulate fluid electrolyte balance. Because they attract fluid and a healthy concentration of proteins in the blood, the cells, and the spaces between the cells helps to ensure a balance of fluids in these various compartments. Moreover, proteins in the cell membrane helped to transport electrolytes into and out of the cell. Like lipids, proteins can bind with carbohydrates. They can thereby produce glycoproteins or proteoglycans, both of which have many functions in the body. The body can use protein for energy when carbohydrates and fat intake is inadequate. The body can use protein for energy in emergency situations. This only occurs when carbohydrate and fat intake is inadequate and stores of glycogen in adipose tissue become depleted. However, since there is no storage site for protein except functional tissues, using protein for energy causes tissue breakdown and results in the body wasting away. Nucleotides. A nucleotide is one of a class of organic compounds composed of 3 subunits: one or more phosphate groups, a pentose sugar, and a nitrogen-containing base. Nucleotides can be assembled into nucleic acids as DNA or RNA, or as the energy-containing compound adenosine triphosphate or ATP. Nucleic acids. The nucleic acids differ in their type of pentose sugar. Deoxyribonucleic acid, or DNA, is a nucleotide that stores genetic information. DNA contains deoxyribose plus one phosphate group and one nitrogen-containing base. The choices of base for DNA are adenine, cytosine, guanine, and thymine. Ribonucleic acid, or RNA, is a ribose-containing nucleotide that helps to manifest the genetic code as protein. RNA contains ribose, one phosphate group, and one nitrogen-containing base, but the choices for the bases of RNA are adenine, cytosine, guanine, and uracil. The nitrogen-containing bases, adenine and guanine, are classified as purines. A purine is a nitrogen-containing molecule with a double ring structure, which accommodates several nitrogen atoms. The bases cytosine, thymine found in DNA, or uracil found in RNA are called pyrimidines. Pyrimidines are nitrogen-containing bases with a single ring structure. Bonds formed by dehydration synthesis between the pentose sugar of one nucleic acid monomer and the phosphate group of another form a backbone from which the components, nitrogen-containing bases, protrude. In DNA, two such backbones attach at their protruding bases via hydrogen bonds. These twist to form a shape known as a double helix. The sequence of nitrogen-containing bases within the strand of DNA form the genes that act as a molecular code, instructing cells in the assembly of amino acids into proteins. Humans have almost 22,000 genes in their DNA, locked up in 46 chromosomes inside the nucleus of almost every cell of the body. These genes carry the genetic code to build one's body and are unique for each individual except identical twins. In contrast, RNA consists of a single strand of sugar-phosphate backbone studded with bases. Messenger RNA, or mRNA, is created during protein synthesis to carry the genetic instructions from the DNA to the cell's protein manufacturing plants in the cytoplasm, called the ribosomes. Adenosine triphosphate. The nucleotide adenosine triphosphate, or ATP, is composed of a ribose sugar, an adenine base, and three phosphate groups. ATP is classified as a high-energy compound because the two covalent bonds linking its three phosphates store a significant amount of potential energy. In the body, the energy released from these high-energy bonds helps fuel the body's activities. When a phosphate group is cleaved from ATP, the products are adenosine diphosphate, ADP, and inorganic phosphate, PI. This hydrolysis reaction can be written as ATP plus water forms ADP plus PI plus energy. Removal of a second phosphate leaves adenosine monophosphate, or AMP, and two phosphate groups. These reactions are also reversible. When ADP undergoes phosphorylation, it becomes ATP. Phosphorylation is the addition of a phosphate group to any organic compound. In such cases, the same level of energy that had been released during hydrolysis must be reinvested to power the dehydration synthesis. Cells can also transfer a phosphate group from ATP to another organic compound. For example, when glucose first enters a cell, a phosphate group is transferred from ATP, forming glucose phosphate. Once glucose is phosphorylated in this way, it can then be stored as glycogen or metabolized for immediate energy. Thank you for watching.